Namaste students. Today is our next topic, that is the double helical model of DNA. When we come to the double helical model of DNA, it is a model proposed by Watson and Crick in year 1953. It is called their model as a double helical model. The name associated is always Watson and Crick. Now before giving the structure of the DNA, they used two background informations earlier done by other scientists like Morris Wilkins and Rosalind Franklin. These were the two scientists. They were dealing with the photograph of DNA. They obtained certain X-ray crystallographic technique and obtained certain photographs. These photographs were used by Watson and Crick to formulate their model. And next was a Chargaff's rule. Now Chargaff was while working with DNA, he found that the ratio of purines and pyrimidines is constant. Like the amount of purine is equal to the amount of the pyrimidines in DNA. So these two informations were used by Watson and Crick to give their double helical model. Now what are the salient features of model? Now first they said DNA is a double helix. Double helix means it has two strands. The two strands are formed of a polynucleotide chains, that is there are two polynucleotide chains in the DNA. Now first of all, the first point was the chains are anti-parallel to each other, that is they have a polarity. Now what do we mean by a polarity in DNA? See, in the last topic we studied about a structure of DNA where a nucleotide has a sugar, a phosphate and a base. This was a nucleotide. The two nucleotides were attached together by a phosphodiester linkage. Similarly, a long chain is formed. Now what happens here? A similar chain is in the opposite direction also. As usual, the base is attached to carbon number 1 only. So now when we see here in a detail, we see that here the 5' end, we call it a 5' or 5 dash, here a phosphate group is free. Like if we imagine this is the last nucleotide, in the last nucleotide the phosphate group is free here and 3' OH end is free here. Similarly, in the opposite chain here this 3' OH group is free here and this phosphate group is free here. So both have opposite polarity. If we indicate it as a 5' to 3', so the opposite chain will be in the opposite direction. So this is called the polarity. We always mention a 5', a 3' and in a polynucleotide chain. Now next, what holds these two chains together? Some bonding should be there to hold the two chains together. Now these bondings are the hydrogen bonds. The hydrogen bonds are present between the bases. The two bases are joined together by hydrogen bonds. Now there is also some peculiar feature between the hydrogen bonds, that A always pairs with T with a double hydrogen bond, while C always pairs with G with a triple hydrogen bond. There is always a purine in front of a pyrimidine. So A always pairs with T and C always pairs with G. The bonds are the hydrogen bonds. Between A and T there are 2 bonds and between G and C there are 3 bonds. So this is how the two chains are held together. Now if we consider the actual structure, now how the DNA looks, it is a double helical. We can imagine it like a spiral ladder case. In a spiral ladder case, the railings are actually the two strands, or we say the backbone. What forms the backbone is the sugar and phosphate. The sugar and phosphate forms the backbone of this DNA model, DNA structure. It forms the backbone. The lines you can see are the nitrogenous bases. Now as we are saying that the DNA can be compared with a spiral ladder case. Now when we try to move up, the first step we keep, let it be facing to the north. Now when we take a complete turn, we again come to a north face. It forms a pitch. A pitch is a complete turn. You can see it is a complete turn. Now the distance of this pitch is 34 angstroms. A complete turn, it forms a distance of 34 angstroms. Now in this pitch there are 10 base pairs. Like there are 10 steps to come again to the north. So there are 10 base pairs. So the distance between two consecutive base pairs comes to be about 3.4 angstroms. This is about the dimensions, and the next important thing is the diameter. The diameter of this DNA double helix is 20 angstroms. Now what keeps a constant diameter? is the pairing between a purine and a pyrimidine. It is never a purine-purine or a pyrimidine-pyrimidine. This will disturb the diameter. So, the diameter is constant because of this peculiar pairing. So, this is about the DNA double helical model. It was given by Watson and Crick, and on the basis of this, he also gave further the semi-conservative mode of replication. And so, this is the ladder case spiral helical model. The two strands forms the railings of a staircase. You can compare it with that. That will be a simple. So, these are the dimensions. Thank you. We will study in further class about the next packaging. Thank you.